So it's always nice to be back in Canada. Some of you may know that I was a postdoctoral fellow not so far from here, from the University of Western Ontario. And my postdoc advisor was Kees van der Wolf, who was Mr. Teda back then. And I worked very closely with Stan Leung, whom I met today. And the reason why I remember all these details is because I have a structure in my brain called the hippocampus, and I have a mechanism here that's called memory that allows me to travel back in time and space and recall those very important events of my life in Canada. Now, what I'd like to do today is to talk about a little bit the broader scale of things than what we have discussed, at least so far. I'm showing you a list of ideas here, or a list of phrases here, and I can ask the question is, how many of you think you are doing something else than that would undergo in this list? And I think the majority of us would say, yes, this is exactly what we are doing. I dedicate my life to one of these things, and so on. Now, this is an interesting thing, is because this is an old text. Actually, it's a table of contents from a book. Maybe some of you remember that book. Is there anybody in this room who would tell me which is that book that has this table of contents? It's a mandatory reading to every single neuroscientist. You may know the name, William James. Of course, this side don't count. Now, this is very interesting, because if you look at this list and phrases, they are all special because they are all about sensation. They are all about perception. They are all about inputs to the brain. And this is not by chance, because William James was very strongly under the influence of British empiricism. And British empiricists inherited these terms from Christianity, and Christianity inherited many of these terms from perhaps Aristotle. So the funny thing is that we inherited this list, and we classify them as expressions with boundaries. And today's cognitive neuroscience is doing mainly a work that is trying to understand these words with the same boundaries as they are boundaries in our cognitive space. And this is, of course, the most ridiculous thing in the world, because how would it be possible that the brain is organized to terms that have been invented by people who had no clue that the brain has anything to do with this? So the reason why I show this, because indeed this inheritance is so strong with us, and most of the agenda that we have is based on this. So here is a typical paradigm that is a descendant of the William James paradigm, which is showing things to the animal, typically a sensory input, and then we are recording something in the brain. Pascal Fries showed beautiful results today. We have done a lot of LFP recordings, but of course both Pascal and I know that those LFP signals are very, very, very important for us, for the observers. But the brain has nothing to do with it. Therefore, it's a relationship between one thing outside in the world and one thing inside the brain, and only the experimenter has access to it. However, when it comes to spikes, we pretend that those spikes, of course, they have to signal something downstream, therefore, that's a true signal for the brain. But the problem with this approach is that the neurons that are being recorded from somewhere here have absolutely no clue what's going outside in the brain. All they see is action potentials from their upstream neurons. This morning, the first lecture by Carlos Schatz was a beautiful demonstration of how the visual system is built, and how the neurons are driven, and how they can sculpture the visual system. But that activity has zero meaning to the animal, because that activity or that pattern or that has zero meaning. It has no meaning. In order to get meaning in the brain, you have to ground to something the activity. It's like when you learn a language or when you hear a language that's totally meaningless, even though there are patterns. You can analyze them any way you want, and you can get millions of clusters, you can do supervised learning or whatever you want, but it's still meaningless. In order to have some meaning attached to it, you have to know another language which you are grounded to. So The only way I think that could work is that the neurons somewhere here should have access to a grounding signal. And my grounding signal is action. So let's start with this, with this cartoon. And let me start with my first axiom. My first axiom is that the most important goal or function of the brain is to predict the future. The reason why brains are predictive devices is because we live in an environment with statistical regularities. And those statistical regularities can be exploited one way or another. On the left side is a very simple organism, could be you can call it the aplysia. What happens in a simple animal like this, it generates an output, generates an action. The consequences of generating an action is that the sensors are moved around, and the consequences of that action are registered with the help of the sensors, and the animal knows that there was a consequence of my action from within. Now, the entire goal of this is, of course, to predict the future, but this aplysia kind of thing, or a simple thing, can predict the future only in a very simple environment and at a very short time scale. So what happens over the course of evolution is that we add a loop and another loop and another loop and keep adding loops to this very simple organism. And the entire goal of this complication is nothing else, just the same thing, is to predict the future, but now predict the future in a more complex environment and at a much longer time scale. So we apply for graduate school because we hope that someday we will become a professor. So this is all cool, but I think the most interesting part is what happens a little bit later. With training, either phylogenetic training or ontogenetic training, the loops become very, very, very smart. And at one point, they are so smart that they no longer need the environment. And this is what I call the disengagement of the brain from the environment. And this is the situation when we can do things like test what-if scenarios. What if I go to medical school? What if I go to graduate school? Without actually trying both of them, without acting them out. Basically, what is happening is that we start viewing the computation of the brain from within without needing anything from the outside world. So in the rest of my talk, I will just try to emphasize this by showing 3 examples. So here's my first example. Everybody knows that baby kicks are very, very important. The Apgar scores correlate with a lot of things, including your IQ and your strength of the handshake and a lot of very interesting things. But what we don't exactly know, or what we didn't know for a long time, is why is it good for the brain? Luckily, you can study this in rodents also, because rodents are, when they are born, especially rats when they are born, they correspond to the transition between the 2nd and the 3rd trimester of pregnancy in humans. And what you see here, there are these baby kicks also, this very reminiscent of what happens in human fetuses. And again, you can ask the question is, why is it good to have these? The truth is that this is not what we ask. What we asked ourselves is if we wanted to investigate what is the first organized pattern that emerges in the brain after birth. So we put in electrodes and we were watching things, and then that was the first time I actually paid attention to it, and we have seen these little clicks. So what happens is this. There is no activity whatsoever. It's like in a premature baby. There's no activity, 0, 0, 0, and all of a sudden there is a big recognizable pattern, and this is one pattern, this is another pattern, and another pattern. If you look at the multiple unit activity, you can see that they are associated with, there are very few units, few action potentials in between. Simultaneous recording of the movements by various sensors, you will see that there is a nice relationship between the occurrence of these patterns. If you zoom in, interestingly, what you will see that this is an interesting pattern, and everybody who knows enough about physiological patterns in the brain and know where we were recording from. We were recording from either the thalamus or the somatosensory cortex. They would say, oh, this is a sleep spindle. Except that this is not a sleep spindle because it happens all the time on all behavioral states, and every one of them is initiated by movement. So it is indeed, it works like a Newtonian brain initially, It is indeed works like the cartoon that I showed you with Hubel and Wiesel, that the brain is there waiting for something without an outside world. It doesn't do anything. It has to be energized. But over time, what happens, of course, is a little different. So at this early age, after birth, the brain is organized mostly vertically, which means that the IA fibers from the spinal cord are going up to the thalamus. From the thalamus, the thalamocortical fibers are already in place, but the long-range connections, the horizontal connections are not in place. And to make the long story short, the good thing about this is very reminiscent of what we heard from Carla Schatz here in the visual system, that this gives you an activity, a spontaneous activity that allows you to learn something about the statistics of not the visual world, in this case, your body space. So we have about 600 or so muscles, and those muscles are twitching irregularly because they receive acetylcholine stochastically, and then they send the signals to the cortex, the thalamocortical cortex, and they generate these isolated spindles completely independent from each other. It's not very useful. However, in the real body, these muscles are anchored to the skeleton. As a result of this, the degree of freedom, that is their independence, is reduced tremendously. And only those neurons only those neurons will fire in the somatosensory area whose representations in the periphery happen to be relevant in space. And the agonistic muscles and the antagonistic muscles will be happening at about the same time. And as we heard, things that fire together, fire together. This is not only, as I mentioned, only in the rodent brain. This is exactly the same thing happens in the human's brain. This is a premature baby that we recorded with Rustam Kazipov. And then you can see there is nothing in between here. And then you see this spindle activity. So this happens only for an immature brain. But as the brain matures, the long-range connections come in, the subcortical neurotransmitters that Sylvain just talked about are very important, and then the EEG patterns and the neuronal patterns relative to each other become adult-like in about 2 weeks. And by the time the spindles are no longer relying on any external sources. They just become sleep spindles that occur spontaneously during sleep. However, this link between movement and spindles never disappear. If you go home tonight and you go to bed and you will fall asleep, sometimes you have a startle reaction. You almost fall out of the bed. It happens to many of us. And if you would allow me to record from your brain, then I would demonstrate you that there would be a spindle that would be triggered here. But my first point was that many of the patterns that are initially dependent on the environmental signals or from body signals, in this case, they can be internalized and they become spontaneous. They disengage from the outside of the brain. Okay, so my next two examples are coming from the navigation system. There are three pillars of navigation. If you would like to use a map, all you need is a map. Of course, you come out from the New York subway or in Toronto. The first thing you would like to do, of course, is orient the map. You have to have an orientation, otherwise you start going up-down instead of downtown. So you need a head direction system. You also need to locate where the subway is on the map, and this is good to have the grid system, which is typically a rectangular system in regular maps. And for that, you have the grid cell system. And the third thing that you have to have is locate yourself. And there is one more thing, which is not shown here, is to have a calibration of the map. Because if you don't exactly know how far the things are, where you have to go, you don't know whether you can take a cab or you have to walk for 5-6 hours. So the reason why I start with the head direction system is because it's a very robust sense. You know, the Aristotelian senses are just a few of them, but in fact we have many more senses, and one of the most basic senses that we have is in fact the head direction system. We didn't even know that there is such a thing until Jim Rank discovered them in 1984. So what is it? It's the simplest possible sensory sense. Why am I saying that? Well, every other sense has complications, such as vision, has color, orientation, selectivity, and all sorts of things. So when you recode from a neuron, you always have to make a lot of controls because they may be coding for something else than just one particular thing that you're looking for. The same thing, of course, with touch and so on. The head direction system cares about nothing else, just direction. So if I'm looking at that particular direction, there are always cells in my number of structures that I will talk about in a second that will fire when I turn around my head. And for each of the 360 degrees, there will be a bunch of cells that will be specific for that particular direction. And you can take me in another room, the entire head direction system will be reset, but the relative relationship of the different neurons to each other will be identical. So in order to examine this a little bit more, we have to recode from head direction neurons, but those are usually hidden in small structures, such as the enterodorsal nucleus of the thalamus, which is tiny, tiny structure. If you would like to know how these neurons relate to each other, you have to have tiny recording electrodes. That's why we are using the silicon probes. Then you can record more than 2, sometimes more than 20 of these neurons. So just to familiarize what I already told you about the head direction cells, if you are able to record from let's say, the enterodorsal nucleus of the thalamus, which is one of the most prominent places for finding head direction cells, or the posterior subiculum, then you can see that each of them are very, very unique and very sharply tuned neurons. Now, there is a gentleman that some of you may remember, his name was Sherrington, who said that if something is important for survival, if something is important for controlling the body, it has to be represented in multiple places and by multiple mechanisms. And this is true, absolutely true for the head direction system, because they are present in the brainstem, they are present in the mammillary bodies, the enterodorsal nucleus, the posterior subiculum, the entorhinal cortex, the parietal cortex, even the lateral septum. So there are in various places. They have very sharply tuned patterns. They are ubiquitously pretty much the same. So the reason why it is interesting, as I said, it's a simple system, and it is believed to be driven by various cues. We don't exactly know what they are, but we know that the visual input is very important, because if I'm looking at this direction and you move the visual world, then my head direction system starts moving. But if you switch off the light, no problem. The head direction system survives. So then it turns out that there might be the vestibular system or neck muscles. There have been a lot of speculation, but the overall idea is that this is a sensory system. Sensory systems are driven by cues, by inputs. That's an interesting statement, because then you can ask, what happens in the absence of sensory cues in those sensory systems? I just recently asked, how would the neurons behave in the visual cortex if there is no vision? Or what happens in the visual cortex, for example, during sleep? And it turns out that as of today, 2017, there is no chronic recording in any species ever done in V1 or V2 during sleep, for example. So we ask the same question here in the head direction system, is what happens if we turn off all the signals that exist that can drive the head direction system? And the answer is very surprising. So in order to answer that question, first what you have to do is to record from a bunch of neurons, as we did here from the head direction system. With LEDs, you can record from the head direction of the animal, and you can use this population vector to reconstruct the head direction from the population firing of the neurons. And you can see that the reconstruction is very precise. All you need is a few dozens of neurons, and everything is just perfect. But we ask, what happens with this head direction system vector when the animal falls asleep? So, in fact, it was the other way around. The animal was asleep and it woke up. But what you can see here is that when the animal is in REM sleep, the neurons are not randomly firing, but they have the same sequence, the same order as before. And you don't see this pattern very well here. But in fact, the order of the recruitment is pretty much the same during REM sleep than in the waking. And you will see it's pretty much the same also during non-REM sleep. So what am I asking here? What I'm asking here is that what is the relationship of this multiple neurons. If one is recording, or one is coding for 30 degrees, the other one is 40 degrees, the other one is 50 degrees, 60 degrees, 70 degrees, will this relationship be preserved when the animal is in REM sleep or non-REM sleep or not, or it's completely random? And the answer is they are very strongly conserved. The difference is that during non-REM sleep, the head direction is turning around 10 times faster. During REM sleep, we can predict which direction the animal is looking at and which direction the animal is gazing. Of course, the head is not moving anywhere. So this is a virtual gaze. So let me show you some examples or statistics to support what I just said. We can take two neurons that have complete or almost complete overlap. So if I'm looking at this direction, the two neurons are overlappingly represent this particular direction. What I see is that it's not by chance that if they are looking at the same space, they are together in time. And in fact, this is what happened. The cross-correlograms, you can see that the cross-correlograms are very, very good. The cross-corelograms are also very good during REM. So these two neurons, not only in the waking, but they keep their relationship between REM sleep and slow wave sleep. The difference is, you can see the time course here. This is 2 seconds, and this is 20 seconds. Now, these neurons have an opposite representation. No wonder their cross-corelograms are negative to each other. So if some neurons are positively correlated, some neurons are negatively correlated, then there should be a transition of no correlation. And the interesting thing is that no correlation happened to be at about 60 degrees. And now this is shown here. This pair of neurons is shown up here, and there are about 400 pairs of neurons, and you can see that indeed the pattern is true for the entire population. Now, the 60 degree, of course, is an interesting thing here. I didn't emphasize, but you already, I think most of the students and postdocs are very familiar with the grid cells, that the grid organization is a hexagon, and that has 60 degrees. And there have been many, many attempts to identify the mechanisms of the hexagon generation, and that was initially the entorhinal cortex, but it turned out that the prosubiculum and the parasubiculum, which have very different structures, they also have the grid. Now, Tim Behrens, using fMRI, they also found head direction cells in the human prefrontal cortex and so on. So it's probably not so special to the entorhinal cortex, but we also know that without this head direction system, there are no grids in the entorhinal cortex. So the possibility is that not only that the head direction system drives the grid cell system in the entorhinal cortex, but the 60 degree may be actually inherited from here. So again, once we have this, then we can ask the interesting question is that these neurons in the postsubiculum, all they know about the world, of course, is the neurons that are, the activity of the neurons that are upstream to that. So with this information or with this idea in hand, we can ask, can we predict the timing of the action potentials in a downstream postsubicular neuron, knowing the population activity up here? And the answer is, yes, we can, and using a particular method. But once you can, then you can ask, what is the best time window within which you can make the prediction? And you can say, in which state you can make the prediction better. What is the best state where you can make the prediction? And it's obvious that in the waking brain, of course, it's much better because we have access to the entire sensory system that is driving the head direction system. So this is the waking one. But the take-home message of this is that when we don't have any information about the external world, we still have quite a large ratio of available predictability. So why is it good and what does it serve? When I turn my head around, there can be a block somewhere, I can close my eyes, I can still interpolate. I can also extrapolate. So this is a way how this system that becomes internalized, it's capable of using the system because of its richness to do the necessary interpolations and extrapolations that are needed for every sensory system. So this was my second example where internalization happens over the course of ontogeny and perhaps a little bit inherited from phylogeny. So now let's go to a more complicated thing, which is the place cell system. Place cells are active from one after the other for at least two fundamental reasons. The first fundamental reason is that what our hero, John O'Keefe, would suggest is that the environmental cues are acting upon the brain, which in turn somehow from visual somatosensory system will reach the hippocampus. And the reason why P1, place cell 1, place cell 2, place cell 3 are firing one after the other is because the sensory convergences are driving them one after the other. In other words, they are driven from the outside world. So this would be a cartoon for that. However, if this would be the entire story, then I wouldn't be able to give this talk without any external cues. I'm capable of giving this talk by myself here without much help, even if I wouldn't look at my slides, is because there is a group of neurons that was initiated when Kari Hoffman introduced me. And that group was active for a while, and that group activity was given to another group of neurons, and another group of neurons, and another group of neurons. And this chain of activity, of spontaneously changing cell assembly sequences in my brain can go forever. So that would be another source of generating sequences, which I call internally generated sequences. And of course, this is the essence of everything that we call cognition. So how do we go about this and show some proof for that? So hopefully the video will work, or the sound will work now. This is a familiar pattern that many of you have seen many times. This is a particularly nice CA1 pyramidal cell that is a place cell. We can change the firing patterns into color. This is the tuning curve of the neuron or the place field of the neuron. But you can also listen to this neuron and you can hear that it's actually firing rhythmically. And it fired at 7.7 hertz. And on this particular tile, and you can see that the rhythmicity of this neuron is very nice. If you compare the ongoing frequency of this neuron with the underlying local field potential, which is nothing else but the sum membrane potentials of all the surrounding pyramidal cells, that frequency is about 7 hertz. So we have 7 hertz and we have 7.7 hertz. It's a non-integer relationship between the two. It's an irrational relationship. The consequence of this is interference. If you have two oscillators, you always get interference. And if you have interference, you've got temporal precision. The temporal precision is very, very good. And John O'Keefe's second discovery showed that this is a very exquisite or interesting one, because if you see the phase of the theta cycle, and this is the animal's position on the track, what you will see is there is a relationship between where the neuron spikes in the particular theta cycle and where the animal is on the track. In other words, there is a relationship between position and phase. And John O'Keefe used this argument to support his theory that the hippocampus is a cognitive map. The hippocampus is a spatial map that helps the animal navigating in the environment. Now, there's a slight problem here, is because the rhythm, the theta, is self-generated, is self-organized. It's inside the brain, whereas the positions are outside in the world. How the two could be in sync with each other. And this is where I became very much interested in the story, because time entered into the field of a static map. So then, of course, you can ask other questions, is that, there are other kind of dynamics, such as the speed of the animal can have some influence on this, and it really does. So what happens if the animal is running twice the speed across the place field, then you can see in this case that during slow run, it takes about 10 theta cycles, 10 cycles to go through the place field. But if the animal is running twice as fast, then it takes only about 5 theta cycles. Now, what happens with the resolution of the environment? And the answer is not much, because if you count the number of action potentials, it turns out that the number of action potentials emitted within the place field are independent of speed. You can go one step further, and then you can ask, what is the oscillation frequency of the neuron? This is an autocorrelogram of the neuron, the oscillation frequency of the neuron when the animal is running fast and slow. And of course, what happens is that when the animal is running fast, then the number of action potentials remain the same, which means that per theta cycle, the number of action potentials doubles, which is a reflection of the stronger depolarization. So it means that the temporal shift or temporal drive or temporal phase precession is in fact much, much faster. So if we accept this, then we can say, we have something here that you can call phase, or we can call it time, because you can measure this one. If you have time and the brain has access to speed information, and now we know that the hippocampus does have speed information, then it automatically gives you distance. So the reason why there is an explicit relationship between position and theta phase is not because the environmental cues are telling the brain by action potential by action potential where you have to fire, but because this is fundamentally a temporal phenomenon, a temporal dynamics, which with the help of speed will give you distance. So that's one step, that indeed, one indication that there is such a thing as internal localization. Now, it still doesn't get us to the problem of memory. But good theories are good theories because they are predictable. Good theories are good theories because they generate testable hypotheses that can be rejected. So the most important prediction of the mapping theory is that if somehow we can freeze an animal here and now in time, and by some magic we can sustain or maintain the theta oscillation, then there should always be a subset of neurons that will signal the here and now, that is, they should fire forever. And those neurons that happen to fire, they should always fire at the same phase of the theta cycle, because that tells you the here and now. So we tested this idea with Eva Pastakova by indeed freezing the animal in a funny way. So we trained the animal in a hippocampus-dependent spontaneous alternation task. The only variable we introduced is that during the delay period, we asked the rat to face always in the same direction and run always at the same speed. So in our power, we made sure that the environmental signals and the idiothetic body signals remain constant throughout the waiting period. The prediction, of course, is that we should find some neurons that will fire throughout the run. As long as the animal is running, there should be a maintained firing forever. And of course, this is not what we found. Instead, we found this. So this would be one neuron, another neuron, another neuron, another neuron. And these are different trials, and you can see that instead of firing throughout the 15 seconds of run, this neuron only fired for about 1 second at this particular moment in the run. This is another one is differently, and then we concatenate them into session averages, and these are the simultaneously recorded neurons. Then you can see that every part of the journey is represented by at least represented by some assemblies of neurons. And the interesting thing is that the lifetime, that is how longer the neuron is active, is similar on all neurons if you are recording from the dorsal hippocampus. And they are the same durations pretty much when the animal would be navigating in the environment. In fact, I can give you data set to anybody who's knowledgeable with analysis of place cells, and you won't be able to tell me whether the animal was running in a wheel, in a memory task, or the animal was exploring the environment. So this is exciting, but not so exciting. The real excitement is that you can say that, this trajectory is valuable. This trajectory is nothing else that you start an activity somewhere in this n-dimensional matrix of the recurrent system in the hippocampus. The hippocampus is a one giant single module that has the largest recurrent activity in the CA3 system of the brain. And then this activity packet goes in from some place to some other place. And that this trajectory is pretty consistent from trial to trial if the initial condition was the same. Now, the good news is that embedded in this trajectory, there is information about the past experience, that is where the animal came from, and the future goal, that is where the animal is going. All we have to do is to separate the activity of the neurons, whether the animal is going to the right or going to the left. So I'm going to show you one example. This is one neuron. You can hear that the neuron is fairly active. Second trial is pretty silent. Active, then you can already make a prediction that now the animal will probably go to the left. It goes to the right. So we already learned the pattern. Now we know what should happen. Then it will come this way. And according to my clock here, I have already talked for 1 hour and 30 minutes. How much time do I have? Okay, so we're asking what's happening now. The animal, of course, have to go to the right, according to the silence, right? If you are still awake, then this is what you predict. But you can see in a second that something strange happens, which is there were 3 action potentials, and then another action potential, and now we start to hesitate to know whether we believe the behavioral description of the animal or behavioral prediction or the prediction of this neuron. According to the neuron's prediction, the animal has to go to the left, and that's what it does. So the animal made an error, but the error could be read out pretty easily from the firing pattern of the neuron. So this is an amazing neuron. This is this neuron, but it's absolutely useless to guide behavior. It's very useful for us to publish papers because we have a beautiful correlation, but it's useless to guide behavior because it fired only for about a second at the beginning of the run. So in order to make it useful, this neuron or this neuron who's member of the similar assembly has to give this information to another group of neurons, another group of neurons, and there should be a chain of activity that goes on in order to be useful at the time when the animal has to make a choice between left and right. And in fact, there are many of these. So what you see here is that there's the same neurons. This is neuron 1, neuron 2, neuron 3, and so on, neuron 1000. And this is the same set of neurons, except that now they are separated depending on whether the animal will go to the left or will go to the right. And it doesn't require much statistics to see that if you take a slice of time anywhere, and if you have enough number of neurons recorded simultaneously, that you can predict with high precision whether the animal will go to the right or will go to the left, 20 seconds ahead, including errors, with about 90% accuracy. So, concluding this part, the interesting thing, of course, is that, if you have this mechanism that allows you to generate sequences, that the default pattern of the hippocampus is sequence generation. The hippocampus just can't help. It always generates sequences. There are so many sequences, but many of these sequences are useless. We have to attach meaning to it, and in order to attach meaning to that, you have to have experience. The experience is what gives meaning to the sequential activity. Now, if something happens during your internal generation, such as somebody interrupts my talk, then my trajectory will jump. And if there will be another question from somebody else, then there will be another jump, and so on. There will be many more jumps, and then all of a sudden, I will see that the upstream, that is the external environment, will control the sequential activity. So indeed, the hippocampus can generate the sequencing in two fundamental ways, but it's not only one. It's not only the external environment, but also the internal environment. And this is the fundamental thing for cognition. This is something that is superimposed on it. So there are two lingering issues that are not so important for the big idea or for big theory, but for the mechanism. So I said that the lifetime of the neurons is pretty much the same. They last about 1 second, which is about 7 theta cycles or so. And within each theta cycle, there are about 7 assemblies or so. And my good friend, the late John Lisbon, attached a theory to this because of the 7 plus minus the gamma cycles that we have shown in the theta cycle, that indeed that may be a shift register, that may be a kind of a short-term memory. So where do these little shifts come from, and where is the lifetime coming from? So I give you the answer. The lifetime, that is the duration of the cell assembly comes from short-term plasticity. Now, measuring plasticity, that is measuring synaptic weights or synaptic strength in a freely moving animal is not trivial, because you cannot record simultaneously easily from connected pairs, but you can do something what we call a proxy of that. And not necessarily between pyramidal cells, but between pyramidal cells and interneurons. So these are from a large number of neurons. We selected a pair of neurons where the presynaptic neuron is a pyramidal cell, the postsynaptic neuron is an interneuron, and you can see that there is a high probability that when the pyramidal cell fires at a short latency, there will be a spike in about 1 or 2 milliseconds. This is a monosynaptic connected pair, and this is called spike transmission probability. Spike transmission probability in a freely moving animal can be used as a substitute for measuring synaptic connectivity and, to some extent, synaptic strength. So with this in hand, we ask what happens when the animal goes through the place field. So this is the place field, and we divide it artificially into 5 bins. Here I said, what is the strength between the pyramidal interneuron connection in the first segment and the second bin, or the third bin, and so on. Now it turns out that we found 2 types of disconnectivities. One that you can call a depressing type, and the other one is you can find a facilitating type. Now we called it parvalbumin-like neurons, the postsynaptic neuron, and somatostatin neurons for the reasons you will see in a second. So this is quite amazing, because what you will see in this pair is that, and you can see it also in the statistics here, is when the animal goes into the place field, what happens is that the place field is very powerfully enslaving those interneurons that are perisomatic, and they are so-called bistratified cells that are controlling the inputs from the CA3 collateral system. In other words, the CA3 collateral system at the beginning of the journey is blocked. Now, when the animal goes through the place field, this blockade is getting weaker and weaker. And if you look at what happens at the perforant path input, which is reflected by this OLM type of cells that are controlling the distal dendrites where the perforant path comes in, initially the perforant path input is open. And the neuron from the distal dendrites can be driven by way of the entorhinal cortical input. But as the animal goes through the place field, that this synapse is getting, this connection is getting potentiated, and the gate will be closed, and the entorhinal input will be weaker. So there's a nice interleaving mechanism here that the initial part of the place field is cortical initiated, but the rest of it is internally generated, internally maintained with the help of the CA3 system. We have done it in different, this was in a rat, so now we have done this now with optogenetic identification in a mouse. There is an example. Instead of, this is a head-fixed mouse where the animal is running on a conveyor belt, and this is about 150 centimeters long. You can see if you record from a large number of neurons, then every part of the journey is nicely represented by some kind of place cells whose lifetime is pretty much the same. and on average is pretty much the same. And then what we can do during the different parts of the journey or different trials is we silence either the parvalbumin neurons or the somatostatin neurons. And you can see also this reciprocal relationship here. Now we are not measuring the spike transmission probability, but we are looking at the firing rates of the neurons, of the place cells, when either of these populations are shut off. So what happens when the parvalbumin neurons are silenced, then initially there is a difference between the controls and the optogenetically silenced situation. The firing rate of the place cells go up, but as the animal goes through the place field, it goes down. And the opposite thing happens when the somatostatin system is closed. So short-term plasticity between the pyramidal interneuron connection is probably responsible for the lifetime of these assemblies. And we have found a similar situation in the prefrontal cortex and with similar outcome, although we didn't do optogenetic experiment there yet. So the second issue is that what is responsible for this internally organized pattern. It's not the miracle that when the animal is going through one place to another, that neurons are sequentially firing after each other. But it cannot be explained why this sequence activity is the same within the single theta cycles. It's repeatedly the same. It has to be some temporal organization inside. And again, the answer is inhibition, and this perisomatic inhibition. Just to make the long story short, what you find here is that this is that hippocampal theta cycle is shown twice. Theta oscillations are oscillations, but not all oscillations are synchronizers. So some of you may remember the early days of the 40 years discoveries and discussions that, synchrony is not a good thing because the entropy decreases and then the storage capacity of the system decreases and so on. Therefore, oscillations are not particularly good. But in fact, this is not a synchrony. You can call it heterochrony or some other names, because the antithesis of synchrony is not necessarily asynchrony. This is a beautifully organized pattern. It's like playing a piano or playing a musical instrument. That's not synchronous or asynchronous either. That's a pattern. So this entire pattern is, the entire phase space of the theta is filled with cell assemblies. And as a proof that indeed the parvalbumin interneurons are very important for that, when we silence them, then all of a sudden the neurons that fire at the late phase or early phase go to the trough. The neurons that fire the late phase also go to the trough, and the neurons fire indeed synchronously now. And I assume if we had enough power and we illuminated a very large part of the hippocampus, that all this thing would be super synchronous, and then we would be talking about epileptic intellectual spikes. So the interpretation of this is that this is information. The downstream readers from the hippocampus must be aware of these sequences. In fact, if not, the behavioral experiments in our hands show that if you scramble this one, then the animal cannot perform a spatial task. And the potential answer to that is because the downstream readers can no longer make sense of this non-sequential activity. So going back to my original picture and original claim, initially, the brain just generates outputs and is looking at the consequences of its action. It's predicting the future in a small organism that is capable of doing it in a very simple way, but then the loops are becoming smarter and smarter, and then we can make the prediction more precise, even in a noisy environment in which we live. And cognition probably happens at a time when the brain is capable of disengaging itself from the environment. Now, I have to make a very important point here, or very important comment here. I don't believe that things happen the way I described in this sequence. It's not that there is a simple organism that completely relies on the external world, and there is a complex brain that is mostly doesn't rely at all on the external world. What I'm saying is that these are different ways of separating these ideas from each other in order to have a disciplined explanation and a disciplined approach to understand them. The ratio of the internal and externally organized, of course, changes quite a lot. In this system, it's probably very high, and in this system, it's probably very low. Most of the things that we are doing is depending on non-environmentally driven things, because we spend very large part of our life contemplating about our events that happened in our lifetimes, including what happened to me many years ago when I was a postdoctoral fellow in Canada. I finish here, and of course I'd like to emphasize that I didn't do all this work by myself. There were many, many important and talented collaborators of mine, including Adrien Peyrache, who is now in University of Montreal, McGill University. I apologize. McGill University. Thank you.
Well, before I invite you to the next event today, that was our final speaker for the symposium. And so I hope that you would help me in thanking those at York University, including the York President's Office, Faculty of Health, Faculty of Science, Ron Perlman, for their assistance with logistics and organization, for the Kremble Foundation, our other donors, and of course the Gairdner Foundation. And finally, if you could help me in thanking our fantastic lineup of speakers. Thank you very much. And now we have a poster session and reception for the next about an hour and a half. So this is an opportunity when you can answer all those questions you've been building up throughout the day and have a drink. So enjoy.